Okay, so good evening children. So today we are discussing about the 10th class first lesson, that is the nutrition. So in this lesson, uh, today we are experiment. What is the experiment? It is oxygen evolved in the process of photosynthesis. So that is the experiment name. So in this experiment, we are writing like the aim, materials required, procedure, precautions and observation. So aim, materials required, procedure, precautions, observation. Five main sidings are there. So in that first one is the aim to prove the oxygen evolved in the process of photosynthesis. So what is the aim? To prove the oxygen gas evolved in the process of photosynthesis. Means half of the question we can write like the aim. Half of the question we are writing like the aim of it is. So that is very simple. And second one is the materials required. Materials required mean here what diagram parts. What are the diagram parts? That is only we are taking like the materials required. Means there is no chemicals here used. We are not using any type of chemicals here. So that's the reason I am not written the any type of chemical materials. Only materials required are there. There is no chemical materials are there. Okay. So what are the materials we need? We need the two beakers. Two beakers we taken. Two test tubes also we taken. Two funnels also we taken, and some hydrilla plants or elodia plants, and one matchbox we need. So now how to start the experiment, or what is the procedure for the experiment? First of all, take the two beakers full of water, and take the two funnels and keep in the two beakers in a reverse. Okay, you see inverted. This is the inverted funnels. So in that funnels already you take the hydrilla plants or elodia plants which is available in our near surroundings. That plant we are taking for our experiment. Means hydrilla. I take the hydrilla plant or elodia plant. Any one plant you take. In the funnels. In the funnels we are taking the hydrilla plants and keeping the funnels in the water, and funnels upside we are keeping the two test tubes. So this is the aperture 1 and this is the aperture 2. Aperture 1 and aperture 2. So aperture 1 we are keeping in the down of the sunlight, and aperture 2 we are keeping in the total dark room. Means 1% also light not enters in that room. Like that dark room we are choosing, and in the dark room we are keeping the aperture 2, and aperture 1 we are keeping the down of the sunlight. So after completing of some hours or at the evening, means morning we are keeping the dawn of the sunlight, and morning we are keeping the dark room, and at the evening. So when you are removing the when we are observing these beakers, first beaker glass tube, and first beaker we can observe the small bubbles are appearing around the hydrilla plant, and there is no bubbles in the dark room. Hydrilla plant, but which is the down of the sunlight. That sunlight down the hydrilla plants, they are surrounded by the what? Surrounded by the small bubbles of it is. Okay, and in this which aperture present in the down of the sunlight. So in that the water also reduced in the test tube due to the filling of the gas. Now slowly remove we are taking the two aperture in one place. At the evening we are taking the two aperture in one place, and first of all we are removing the aperture 1 and aperture 2. Now we are taking the aperture 2 glass test tube slowly with the means we are keeping the finger downside of inside of the water, and closing the finger to the mouth of the test tube, and slowly open the test tube outside. And when you are keeping the one glowing mastic on the or instant stick or glowing mastic on the test tube. So there is no glowing of the mastic and instant stick. There is no glowing of it is. There is no glowing of it is. Okay, and in the first test tube, when you are slowly removing the test tube with the help of the fingers, and when you are keeping the glowing mastic on the test tube from the first beaker, so the mastic glowing very brightly, means it is glowing more. Mastic glowing more of it is, because first test tube releasing the more oxygen or contain the more oxygen. So more oxygen releasing from the test tube. That's the reason the mastic is glowing more amount of it is. Okay. So this is what it is about the experiment oxygen evolved in the process of photosynthesis. Why the first test tube mass tube glowing more and second test tube did not glowing? Because first test tube releasing the more amount of oxygen to surroundings. That's the reason. That's the reason it is the glowing of more of it is. Okay. So means sunlight only useful for the doing of photosynthesis in the plants, and in the presence of the sunlight only the plants are releasing the oxygen gas of it is. So what are the precautions? So precautions are, first point is slowly remove the test tube from the water, otherwise the gas is going to atmosphere. That is the first experiment. So first precaution. Second precaution is keep properly, catch the properly that mastic, otherwise our fingers will burn and we are getting the burning sensation near of our fingers. So that is also second precaution. And what is the conclusion? So from this experiment I understood that, or I concluded that in the down of the sunlight only all green plants are releasing the oxygen gas. All green plants are releasing the oxygen gas of it. Okay. So this is the what it is about the experiment. Thank you.